So this is chapter 3 from Krogh, Molecules of Life, and I'm going to break this into a few different lectures just so hopefully everything is very clear. So the first point to make here is that carbon is absolutely essential to life. Cells are mostly just carbon and water, in fact. There's a bunch of other stuff mixed in there as well, but that is the majority of what makes up a cell, is those molecules, carbon, hydrogen, and oxygen. And carbon is the basic building block of the 4 macromolecules of biology. And those 4 macromolecules are carbohydrates, lipids, proteins, and nucleic acids, or DNA. You could also put RNA into that category as well, but we're just going to be concerning ourselves mainly with DNA in this section. So carbon is so important because it can create 4 bonds. You can link carbon up with 4 different things. And the study of carbon compounds or carbon-containing compounds is also the basis of organic chemistry. And as kind of an aside, when people say organic, if they're talking about groceries, they're meaning something entirely different than when a chemist or a biologist says organic. So organic in scientific terms means carbon-containing compounds. So why is it that carbon is so important, and why is it that it can create 4 bonds? So carbon's atomic number is 6, which means it has 6 protons. Typically it has 6 neutrons, and a neutral atom of carbon will have 6 electrons as well. So let's go ahead and add in our electrons. So we put 2 there on our first shell, and then we have 4 more left to go, right? So we'll add those 4, and now we have our electron diagram of carbon with 2 electrons in this innermost shell, and then 4 out here along the perimeter. And you should remember that atoms are going to want to fulfill that octet rule. And what I mean by that is they're going to want to fill up their valence or outermost shells. So the way that carbon is going to be able to fill up its valence or outermost shell is to make bonds with 4 other things. So as you can see here, the fact that carbon can make 4 bonds allows it to create these large molecules. And remember, a molecule is just a bunch of atoms all stuck together. So it can make these carbon skeletons which vary in length. It can be branched or unbranched. You have this branched one here. This is isobutane. It can form double bonds. So carbon can form double bonds with each other, and those double bonds can change location. So this double line here is representing a double bond, means there's 2 electrons being shared here. This single line here is representing a single bond, which means 1 electron is being shared here. And you'll notice that these have the same chemical formula. So you've got 1, 2, 3, 4 carbons, and 1, 2, 3, 4, 5, 6, 7, 8 hydrogens. The same thing is true over here. You have 1, 2, 3, 4 carbons, and 1, 2, 3, 4, 5, 6, 7, 8 hydrogens. And so this is a topic we're going to talk about in a few minutes. This is called an isomer, but just notice here, carbon can form these double bonds, and they can be in different spots. And that actually affects how this molecule is going to behave, is where those double bonds are located. You can also form a carbon skeleton with rings. So you get this nice ring here, and like in the case of this benzene, you can have double bonds in these rings as well. And a carbon that is formed into a ring is going to have different properties. I should say a carbon skeleton that's formed into a ring is going to have different properties than one of these linear or branched carbon skeletons, or even one of these ring skeletons with a double bond. So, like I was saying, they can be large and varied, they can be linear, they can be branched, they can be bonded to carbon atoms themselves. So carbon can be bonded to carbon, or it can bond to other molecules or other atoms. And this idea of a hydrocarbon It is the simplest organic molecule. The example here is methane, or CH4. Gasoline is also a derivative of a hydrocarbon. And a hydrocarbon, as the name implies, is just hydrogen and carbon put together. And every single hydrocarbon has this sort of unique 3D shape, which gives it its various properties, depending on whether or not it's branched, depending on whether or not it's making double bonds, depending on whether or not it is forming a ring. Okay, so quick review. Carbon, an extremely important molecule in chemistry and biology. The reason for that is because it is able to form 4 bonds, and it does form those 4 bonds very frequently. It can also form double bonds, and those double bonds can be in different locations. It can also form rings, and what kind of shape your carbon skeleton forms is going to determine its properties. It's going to determine the way that carbon skeleton behaves. And they call them carbon skeletons because they kind of look like skeletons. So you also have different ways of representing different molecules. This is just in the context of carbon, but you could use this for anything. This is probably one that you've seen before if you've taken like just general chemistry, and it represents the carbon sharing a electron with hydrogen, and the same thing for all these 3. It's just carbon sharing electrons with hydrogen. You also have the ball and stick model, which gives you a sense of CH4's 3-dimensional shape, but you could do it for anything. But this gives you a sense of the actual dimensions of it rather than having it be this 2-dimensional representation. And you have the space filling model over here, which shows you a bit more about the size of these atoms and sort of where their electrons are going to be hanging out. So here's a space filling model of a water, and it's just showing you that the electrons are spending most of their time around this oxygen, which we're going to talk about in just a second here. So you also have functional groups. Functional groups are molecules which are attached to hydrocarbon skeletons, and these are things that are participating in chemical reactions. So you have things like a hydroxyl group, or a carbonyl group, or an amino group, or a carboxyl group. And these are all found in various carbon-based molecules that you'll find. So here is just a picture of a few of these things. You got your hydroxyl groups, your carbonyl groups, which is this carbon double bonded to an oxygen, this amino group, which is the nitrogen, and your carboxyl group with it, which is a C double bond O single bond OH. And if this is freaking you out, it's okay. I'm not going to ask you guys to memorize functional groups. That's not really what this is about. If you take majors biology, then they'll probably make you do that, but not me. I may ask you some questions about some of these things, like, well, what does a functional group do? Why do you have a functional group? Something like that. But I'm not going to say, like, okay, put a circle around the carboxyl carbon or something like that. So there are different ways that these carbon skeletons can be synthesized or built up, and there are different ways that they can be digested or break down, because macromolecules are quite large, and they often consist of these polymers, which are the same repeating units. So a polymer is just a repeating unit of a monomer. So a polymer is just a repeating unit of a monomer. So when you have dehydration synthesis, also sometimes called condensation synthesis, but I'll usually refer to it as dehydration synthesis. This is building something, and what happens is the different atoms get stuck together and they wind up dehydrating. They wind up losing a water atom, a water molecule. Sorry, there is no water atom. They wind up losing a water molecule. All right, so things are typically built up via this dehydration synthesis. Certainly not the only way, but it's a very common one. You also have digestion, which is aka hydrolysis or water breaking. And what's happening there is that water is coming in, and the components of water are being added to the molecules that are being broken down. So dehydration building, hydrolysis, breaking down. And this is just a picture of that. So you have some monomer here and some monomer here, and it says condensation. That's fine. We can call it that. I'm going to call it dehydration. You can think of these two things as being synonymous, dehydration and condensation. They can be thought of as the same thing. What happens is, in a dehydration synthesis, you have this H and this OH, and they come together and form this water molecule. And the ends that they left on each one of these monomers, whatever these monomers happen to be, maybe this is some carbon atom, maybe it's a polypeptide, maybe it's something else, but the water comes off and the monomers get stuck together. The opposite of this is hydrolysis, and you're actually breaking two monomers apart by adding the components of water to either side. And you can see down here, they are together as a polymer, and then water has come in and broken them up, and you wind up with a monomer over here and a monomer over here. This is just another example of a dehydration synthesis and hydrolysis of maltose, which is a disaccharide. Di meaning two, saccharide meaning sugar. So you have two glucose molecules here. If you remove this water, that you have here, they come together to form maltose. And you've got this water unit out here because it came off. It came off. OH here, H here, came together. You get your water out here, but you stuck these two glucose molecules together. The opposite of this reaction is to take this water and bring it in and put your H here and your OH here and cause these two to split apart. Okay, that is hydrolysis. Okay, that is the breakdown of a polymer. So again, the main thing I want you to get out of here is dehydration tends to build. So when you have dehydration, you wind up getting different parts stuck together and you get some water out at the end. And hydrolysis tends to break down. And with hydrolysis, you have water at the beginning, and you wind up with separate parts, and you've lost the water. Water, you didn't lose it, but it is now attached to these different monomers. So this kind of leads us on to the next question here, which is this question of polarity and dissolving. And the main reason we're going to talk about polarity in this class, there are many reasons to talk about polarity, but the main one for us is because of how it is related to this dissolving process. Because later on, we're going to talk about diffusion and osmosis, and this becomes important. So let's start with what makes something polar. Well, as we talked about in lab, electronegativity is the sort of atom's desire to pull electrons toward it. Actually, I have a slide for it. Or how much an atom wants to hog the electrons that maybe another atom is sharing with it. So this leads to an unequal distribution of charge. When you have one atom that's hogging all of the electrons to itself, it's going to be more negative than the other atoms because it has more electrons. It has more of those negative charges. So here's a picture of H2O, and oxygen is very electronegative. It really wants to hog those electrons. And these numbers represent quantities that are actual electronegativities, but don't worry about those. It's totally fine. The point here being is that oxygen loves to hog these electrons, and so oxygen is going to wind up being quite negative. And it's going to give it negative polarity near the oxygen and positive polarity near the hydrogens. So water winds up being a quite polar molecule because oxygen is hogging all of those electrons. Oxygen is very negative. It becomes a very polar molecule. So molecular shape also comes into play here. So if something is symmetrical, it tends to be nonpolar. So here is CCl4, and chlorine is much more electronegative than carbon. So keep in mind what that means. So if I say chlorine is more electronegative than carbon, chlorine is going to be pulling all of those electrons away. But CCl4 winds up not being very polar because the chlorines are pulling electrons away equally. They're pulling them away from the carbon equally. So imagine, if you will, three people, all of whom have like three pieces of rope that are tied together. They're pulling away, but it's not making a difference because they're all pulling away equally from one another. So why does this polarity stuff matter? And by the way, what I would like you to know is that there are two things that cause a molecule to be polar. That is electronegativity, causing an unequal distribution of charge because that one atom is hogging the electrons, and non-symmetrical molecules cause electronegativity as well. And there's a bunch of trick questions I could throw at you in terms of putting like the same atom, but having it be, asymmetrical. But don't worry about that. My objective is not to trick you with this stuff. So that's that. So the reason that this matters is because of this phrase right here. That is, like dissolves like. So as we talked about a moment ago, water is quite polar. So anything else that is polar will wind up getting dissolved in water. And non-polar substances will not be dissolved. And this is a very big idea. There's a lot of underpinnings to fundamentally what we understand about biology here. Okay, things like your cell membranes that we looked at in lab. Those are non-polar, and that's why they don't actually dissolve in water. So